And to really nail the point, health is not all in your genes, because genes assess the risk of moving into a disease, whereas metabolomics is the proximity to the phenotype, to the definitive representation of those outcomes. And you can take genetically identical models, and this is a preclinical model. For example, black 6 mice are genetically identical. And you can offer them 2 different diets, or create a study with 2 different diets. And twins, these studies have also been recapitulated in humans in terms of specific twins, that you can give genetic identity models different diets, an unrestricted high caloric or high fat diet versus a caloric restriction diet. And with genetic identity, you get very different changes in health outcomes. On one hand, with overnutrition, you get disease and early death, and with caloric restriction or controlling the metabolism or metabolic inputs, you get a long and healthy life. And of course, this is subject to intense investigation looking at the pathways that are supporting this to help promote salubrious and long-term health. And this is in large part, again, microbiome and microbially influenced. And Brian talked about some of the interplay between host genetics and microbial composition in the gut and in other communities in the bodies, and oral and pulmonary. There are other microbiome communities, microbial communities. That a definitive study done by Ron Segal in Israel, the Weizmann Institute, looked at repatriation of the Israeli diaspora back into the Israeli state back in the 50s. And the question was, looking at a relatively homogeneous genetic population of Israelis that repatriated back to Israel, what is the gut microbiome composition in this population? And they found that environment has a much, much more powerful role in shaping gut microbial composition relative to host genetics. So all these things reflect distinct signals and processes from host genetics that come from and can be captured from metabolic identification or profiling.